It is now my pleasure to introduce tonight's speaker, Dr. John Flanagan. John has served on our board of directors since 2017 and currently chairs our research committee and serves on our strategic planning committee. He graduated in optometry and vision sciences from Aston University in the UK, where he later earned his PhD. Upon graduation, he held faculty positions at the University of Waterloo and the University of Toronto in Canada. As a vision scientist, John's expertise in glaucoma, neuroscience, and neurobiology has been tremendously helpful to guiding our scientific programs. Thank you, John. John is the 8th and inaugural Dean and Professor at the Herbert Wertheim School of Optometry and Vision Science at the University of California, Berkeley. In addition to his duties as Dean, John also devotes his time to glaucoma research, and if I can say so, we know the quality of his research is exceptional since he also received a Schaefer Grant for innovative glaucoma research in 2017 for his research on neuroprotection. So John brings many wonderful skills to his work with Glaucoma Research Foundation. We're really grateful to have your leadership and your support on our research committee and our activities. So, ladies and gentlemen, it's my pleasure to introduce John Flanagan.
Well, good evening, everybody. It is such an honor to be here amongst so many friends. One of the great honors of my career has been to be invited to be on the board of the Glaucoma Research Foundation. And I would like to take this opportunity to sort of publicly thank the courage and the foresight of Andrew, Tom, and the board for bringing me in as the first optometrist on the board of the Glaucoma Research Foundation, and long may that partnership continue. So thank you very much, Andrew, Tom, and the Glaucoma Research Foundation. It is my wonderful job tonight to talk to you about the research achievements of the Glaucoma Research Foundation over the last 46 years. The founders of the Glaucoma Research Foundation were John Hetherington, Robert Schaefer, and Dunbar Hoskins, all here in San Francisco. And really, it's such an honor to continue their tradition, what they started. And I hope they're looking down upon us this evening, and that they will see over the next 15 minutes or so how we honor their tradition and honor what they started. The Glaucoma Research Foundation was founded by them 46 years ago, and we are dedicated to scientific research, to deepening our understanding of glaucoma, new treatments, and our aim is to find a cure for glaucoma. And we do that in two ways that I'll tell you about. We have this amazing multidisciplinary collaborative research effort called Catalyst for a Cure that really pioneered collaborative research over the last 20 years. And it's become a model for so many other agencies. I have the great honor of being able to call Randy Shackman, the Nobel Prize winner for medicine, who's a biology professor at University of California, Berkeley. Not only is he a colleague, but we consider, Kathy, my wife, and I would consider him a friend. And he now leads a consortium of researchers looking at Parkinson's disease. And they have the same model. Bring people from different disciplines together, provide them with funding, and let them do their magic. The difference between the Glaucoma Research Foundation and the Parkinson's consortium is about $1 billion. They were founded by one of the co-founders of Microsoft, who had a personal family history in Parkinson's, and they have $1 billion endowment. So if any of you in the room would like to provide $1 billion endowment for the Glaucoma Research Foundation, please see Nancy. She'd be glad to speak to you. But I must say, I do think we take some responsibility for the model that is being so successful in different areas of research and has become a model for foundations like us. We also have my favorite, the Schaefer grants. I was the recipient of a Schaefer grant, as Tom said. The Schaefer grants allow us to take risks. One of the sad things about modern-day academia is that most of us are not really allowed to take big risks. To get federal funding, or federal funding does not like to fund risks. We need small pilot funding models like the Schaefer grants to be allowed to have a crazy idea and run with it and then be able to get federal funding. And that's what we'll see has been so successful with the Schaefer grants. It's led to huge amounts of federal funding coming into glaucoma research. But those $55,000 grants are so successful and so needed within a community, research community, to allow you to take a risk. And so thank you for all of you here tonight who have helped fund Schaefer grants. Our guiding principles are that of discovery, collaboration, innovation, and empowerment. By by enabling discovery, fostering collaboration, we're able to really engage with innovation and empower our researchers to help find that cure. So again, thank you for all the help that you give us. This idea of this collaborative research model, and I really must call out Stephen and Michelle Kirsch, who really helped form this model 20 years ago. Thank you for your great foresight as well in helping us here. The idea is that you take early career researchers and you provide them with funding that they don't have to spend half of their time applying for. And you tell them, we want you in our community. We want you to work on glaucoma. And we find the best young scientists and we give them some of the best mentors on the planet and allow them to work towards finding a cure for glaucoma. That is what's behind this collaborative research model. And it's worked so well that even the National Eye Institute of the National Institutes of Health have now adopted this model. So let's go through a little bit of the history. So 1978, as we were founded, as I say, Dr. Schaefer, Hoskins, and Hetherington. 1984, the first Catalyst meeting was formed to stimulate collaboration with glaucoma and experts in different fields towards glaucoma. 1997, the Glaucoma Research Foundation researchers identified really the first gene in terms of glaucoma. And we were talking about genetics on our table tonight and talking about the wonderful Janie Wiggs in Harvard. And I had the privilege of being at one of her recent talks. There are now 120 identified genes in primary open-angle glaucoma. That's quite remarkable. It's a complex disease. We expect multiple genes, but it really is remarkable the headway that's been made in this area. But the first gene was identified through funding through the Glaucoma Research Foundation. The other thing that was remarkable, 1998, we had the Collaborative Normal Tension Glaucoma Study. This was a landmark study. This study, led by the truly amazing Stephen Drance out of Vancouver and Douglas Anderson out of Bascom Palmer, two giants of our field. And they asked the question, with normal tension glaucoma, is it worth lowering pressure? Is it actually worth trying to get a normal tension glaucoma patient to have lower pressure? It was the first multi-center glaucoma study. It was actually the second multi-center trial in ophthalmology, because the first ever federally funded multi-center trial was actually a trial about diabetes, the ETDRS study. This was actually the second, and it was funded entirely by the Glaucoma Research Foundation. And what we found at that time was that, yes, indeed, that it was worth lowering pressure. This nomogram that was just published a few years ago that showed 80% survival rate in those patients where their pressure was lowered 30% over 5 years versus 40% in untreated. A remarkable clinical trial, completely funded by the Glaucoma Research Foundation. This really set the tone for the quality of research that this foundation has been responsible for over the years. In 2002, that was when we launched the first ever Catalyst for a Cure. That's when the funding from Stephen and Michelle Kirsch kicked in, and we funded this first ever Catalyst for a Cure. And that lasted 10 years, that first group. And in those 10 years, they were the first people to identify that glaucoma was actually a neurodegenerative disease. And that was incredibly important. Not only that, but this group of scientists, David Calkins, who was my predecessor as chair of the research committee here at the Glaucoma Research Foundation, Philip Horner, Nicholas Marsh Armstrong from UC Davis, and the wonderful Monica Vetta from the University of Utah, all of whom are still involved in their different ways with the Glaucoma Research Foundation. Not only did they realize that this was a neurodegenerative disease, they were the first team to also recognize that there were specific windows within glaucoma that the disease could be arrested, so that we could intervene at certain times with great success. They also identified or developed the first mouse model that we've used for years now in glaucoma, the microbead model, as we call it, for mice. Mice are very important in research, in medical research, because we can control the genetics. So yes, you may feel there's not much in common between us and mice, but fortunately there is, but we can't manipulate the genes in higher animal models quite yet. But in mice models, we can manipulate the genetics and find ways of discovery through that process. And they developed the first real mouse model that's still being used today. In 2018, we developed the second Catalyst for a Cure to discover biomarkers. So the Catalyst for a Cure 2 team identified certain biomarkers for change, and this was really, really important. They concentrated on 3 particular biomarkers that are now being used in clinical trials around the world. And in that team included Alfred Dubra from Stanford University, Jeff Goldberg, that many of you know from Stanford as well, Andrew Huberman, who many of you might know better as for his incredibly popular podcast on general science, and Vivek Srinivasan from University of California, Davis. So they really allowed us to think that we could do clinical trials in a much quicker and more controlled way, which is very important in terms of drug discovery. The third Catalyst for a Cure launched to focus vision restoration, and that was in 2019. And I know we have at least one of the team here tonight, if you'd like to stand and be recognized. Jinduan. Jinduan's from University of California, San Francisco. Also Yang Hu from Stanford, Anna Latorre from University of California, Davis, and Derek Wellesby from UC San Diego. This group really started looking at how we could not only protect the retinal ganglion cells that are dying at the back of the eye, but how we might be able to restore it. And this team was multidisciplinary too. They had skills in neuroprotection, basic physiology, cell biology, and in Derek, a clinical ophthalmologist, a clinical scientist, who was also an ophthalmologist. And they looked at restoring vision, transplanting retinal ganglion cells. And they very quickly identified 3 genes that, if we turned it off, would improve the survival of retinal ganglion cells. And also identified genes that needed to be turned on to promote survival of injured retinal ganglion cells. And really, this team in particular has set a new standard for us in terms of discovery. Developed imaging techniques to be able to monitor how the transplanted cells were living and working. They discovered a protein, osteoporin, that protects optic nerve cells and has the potential as a biomarker of glaucoma. And their plan over the next few years is really to assess the efficacy of these promising interventions, looking at optimal gene combinations to maximize cell survival. You know, one of the things we've learned about neurodegenerative disease, not just glaucoma, but all neurodegenerative disease, is that we need to take more of an approach a little bit like cancer, where it really depends at what stage the disease is. It's not necessarily one particular cure, but if we know the stage of the disease, we may be able to help the disease at that point and have a sort of multifaceted approach to the disease, where you might need different aspects at different times of the disease. And this team has really been at the forefront of developing that sort of, that way of thinking about the disease. So they're creating gene vectors that are optimized for human use, and they're already into phase 1 and 2 trials. And their hope over the next 3 years is to really take us into preclinical and then clinical trials. This is amazing. This is what we've all been hoping for. And this is what bringing these teams of scientists together has done. And I think that we really are at a time of great hope. And you're probably sick to death of scientists like me over the years at the podium saying, 10 years. 10 years. Give us 10 years and we'll have a cure. You've all heard that before. But I don't think we've ever been closer to that time where we may be within years away of that sort of clue. Yeah. So, launching into vision restoration. The next really, I think, amazing thing that has happened through the Glaucoma Research Foundation that has kept us at the forefront of discovery is that in 2021, we started a meeting called Solving Neurodegeneration. And we called it the Solving Neurodegeneration Catalyst Meeting. And if you look at the date there, April 2021, no surprise that there's not a venue on that brochure, on that pamphlet. It was a Zoom meeting because we were all within the pandemic at that time. But it was astonishing. The idea behind this meeting was that given that glaucoma is a neurodegenerative disease, let's bring some of the top researchers in the world from all aspects of neurodegenerative disease, Parkinson's, Alzheimer's, glaucoma, bring them together to talk about the potential commonalities and differences between the different neurodegenerative diseases and see if we could help each other as communities to go further ahead. And I must admit, as an eye person, I was left with thinking that the glaucoma researchers might really help the Alzheimer's and Parkinson's researchers. And indeed, I still think we can. But together, we're stronger. And this meeting was sponsored by the Melissa and Frank Theodore Barr Foundation, and great thanks to the Barr Family Foundation. And we did this in partner with the Bright Focus Foundation. And the Bright Focus Foundation very specifically fund research in glaucoma and Alzheimer's. And so it was an obvious meeting of those strengths. Very quickly after that, and a really wonderful review paper was published in a very high-level journal that followed those discussions and published the discussions out of this meeting. And very soon after that, we launched our fourth Catalyst for a Cure to focus on neurodegeneration. And it became the first time where we had two concurrent Catalyst for a Cure groups, CFC3, who were still looking at vision restoration, and CFC4, prevent and cure neurodegeneration. So how did we form CFC4? Four amazing scientists with completely different backgrounds. Santo de Makita from the Mayo Clinic is an Alzheimer's researcher. Melissa Margita from Harvard is a glaucoma specialist. Karthik Shekhar from the University of California, Berkeley, is one of the world's leading bioinformatics researchers and has really pioneered bioinformatics for the eye and has a particular interest in glaucoma, thank goodness. And Humsa Venkatesh is a glioma researcher, a brain cancer researcher. You might think, what on earth are we doing bringing those four together? But the skills they have, there's so much commonality. Bring them together and say, you need to help us find a cure for glaucoma. And that's what they're working on, a remarkable team. Melissa, just to let you know, has had already an amazing research history. She is the one who's found this link with APOE, which is also a common link in Alzheimer's disease. So she's been doing some remarkable work in glaucoma prior to this collaboration. So they're experts in brain vascular biology, glaucoma, computational science, and neuroscience, and exploring those commonalities between the different diseases that they work on. And having been able to witness the development of this team, I said that CFC3 is setting the standards that we've never known. The amount of publications, the way that they're going, looks as though they'll be in clinical trials within a few years. This team has equal potential. We're very, very excited. The other thing we do, I mentioned earlier, is we bring in mentors. And some of the mentors that we have for these two teams are truly the top people in the world. With the CFC4, Shane Littleoe from NYU, and in Alzheimer's research, Sally Temple, who runs a big research center in New York for genetics and stem cell research. So truly remarkable. Adrienne DePolo from Montreal, one of the leading neuroscientists in the world in glaucoma and neurodegenerative disease, who interestingly has found a link with insulin and is currently working with Jeff Goldberg from Stanford on looking at insulin as a neuroprotective pathway for glaucoma. Fascinating work. Here we see some of that CFC4 team working together in their labs. You see how young they look, at least to me anyway. They look so young now. It's great. The other thing that we have fostered over the last couple of years is allowing the CFC3 team and the CFC4 team to regularly meet together and influence each other. And that has been incredibly powerful too. And we now have very distinct collaborations between certain members of the two teams. In particular, Xinduan, who's here tonight, working with Karthik Shekhar from Berkeley in terms of bioinformatics. Very, very powerful collaborations. So this whole idea of the catalyst for a cure, it has influenced the greater world of science, but it is contributing so powerfully for us finding that cure for glaucoma. And here you can see the interconnections between these wonderful teams that have been brought together. So if ever you wondered what your research was doing, your funding, this is what it was doing. And I have to quote Jeff Goldberg, because Jeff is one of the scientific advisors to Catalyst for a Cure 3. And in February, when we had our last in-house meeting of the two teams, and we were doing our review session, Jeff, who is not prone to exaggeration or hyperbole, said, maybe we are finally able to realize that we're only 10 years away from a cure for glaucoma. So let's hope. Last year, we celebrated our 45th anniversary, and again had the joint meeting of the two CFC teams. And we also had our Solving Degeneration 2 Catalyst meeting. Again, this was held in Boston with funding from Richard and Caroline Sloan. Remarkable set of external speakers, 4 speakers from the very top of their field, just gave up their time to spend time with our CFC groups. It was a wonderful, wonderful meeting. Again, a publication will be coming out soon to summarize the findings of that meeting and the progress. And we're excited that, along with last year's, we feel this is such a successful meeting that we'll be doing it again in 2024. Here you see the picture from last year. Small group, but really high-powered, all working towards finding a cure for glaucoma. In 2024, we've got speakers Valerie Wallace from the University of Toronto, one of the world's leading regeneration researchers, Dan Saban from Duke University, Jigan He and Sally Temple, who will be our keynote speakers and mentors for our CFC groups. So again, we're excited to do that. The other thing that we have to celebrate this year, just going full circle, back to the other funding model. I talked about those small $55,000 grants that we give as the Schaefer grants. In 2024, this year, we're giving a record number of those grants. We're able to give 10 of those grants. Thank you. And again, I know there are many of you in this room who have helped fund these Schaefer grants. So again, these are the grants that allow us to take risks. They allow us to do small pilot studies, small clinical trials, really look at those crazy ideas that might just mean something. And there is nothing more precious to a glaucoma researcher than being able to try out some of these ideas. So again, please remember the Schaefer grants when you're thinking about what it is we fund. So they fund these pilot studies, investigations. I'd just like to share a couple of quotes here. Julia Richards from the Kellogg Eye Center at the University of Michigan, perhaps the single most productive piece of funding that I have ever received. It's not a lot of money, but it just helps do those crazy, crazy things. And Kate Keller from Oregon Health and Science University, the Schaefer grant was instrumental in kickstarting my research career in glaucoma. This is what we want. This is how we can help the scientific community. And these are the 10 scientists, and you can see they're from all over the country: Columbia, Indiana, NYU, Stanford, Pittsburgh, Hong Kong, Nigeria, Mass Eye and Ear, and University of Miami. And they are truly some remarkable researchers amongst this group. Let's hope that some of their crazy ideas result in maybe our next Catalyst for a Cure group. So over the past 46 years, the Glaucoma Research Foundation has invested $54 million into collaborative research and has really found groundbreaking discoveries. They've revolutionized how we understand glaucoma, paved the way for new therapies to protect vision, and made promising studies towards vision restoration. What do we have to look forward to? We genuinely have to look forward to neuroprotective therapies. We're right on the cusp. We have to work on drug delivery. If we find the best drug in the world, we have to be able to deliver it where it's needed. So drug delivery and other innovations, additional minimal invasive surgical interventions, vision restoration. So obviously, if we find a cure for glaucoma, a neuroprotective therapy, eventually we won't need vision restoration. But for a whole generation before that, we need to find ways of helping restore vision. So vision restoration remains high on our agenda, and obviously a cure for glaucoma. That's our mission, and that's, with your help, is what we'll be able to do. I am so honored to be the chair of the research committee and help guide some of this research. It truly is an honor of my career to do that. I thank you for the help that you give, and I know that our future is brighter. And thank you very much. I hope you enjoy your evening. Thank you.